I think we have all the technology set up. That was the main challenge.
All right. So thanks everyone for joining us today. I hope you all had a great week. Today we have the pleasure to host Brendan, who is the founder of Deep Genomics and a professor at University of Toronto at the Vector Institute. And he'll be sharing with us some thoughts on how new genomics foundation models are transforming drug discovery. So before handing the mic over to you, Brendan, let me remind you all of some house rules. As usual, clarification questions will be allowed to be asked during the talk. For people in person, if you have a question, just raise your hand and wait for me to give you the mic so that people online can hear you well. And for people online, you just have to type the word question or I have a question in the chat. And when the time is right, I'll let you directly ask it. There will be some additional time at the end of the talk for people to ask further questions, make some comments, or have a more informal discussion with Brendan. That's pretty much it for me. So whenever you're ready, Brendan, where is yours?
Okay. Thank you, Rim, and thank you for organizing all of this. I've heard you're kind of the powerhouse behind all of these seminars, so thank you for doing that. And I'm excited to be here. I think this is the first time I've been in this new facility. I think the last time I visited with you, we were in the old location. So it's great to be here, and just to get to know the audience a little bit. How many of you have built like very large neural nets, like kind of foundation models or deep learning systems or whatever? Kind of raise your hands if you've built those kinds of models. Yeah. How about worked on genome biology, like looked at genome data sets, that kind of stuff? Okay, great. Yeah. Oh, good. And you're getting some votes online. Good. Okay. And how about drug discovery? Like anyone worked on drug discovery? Okay, thanks. Good. All right. Well, wow, it's kind of like a third. So just the right mixture. All right, so let's get going. Basically, what I'm going to talk about is, first of all, our business strategy team and ML hiring plans, because we are looking to hire people in machine learning, both machine learning specialists, people who can build foundation models, also machine learning scientists, people who understand sort of the translation, and other related roles. And so I did want to talk about what Deep Genomics is trying to achieve in terms of its business, because in this field, you think about recursion or think about in vitro, you might be wondering what really is the business model. So I'm going to talk briefly about that just to set the context. And then I will talk about, of course, the main topic here, which is causal foundation models of genome biology and why I think those are quite important. And then I'm going to present a lot of data showing that the systems we built and others have built as well are the real deal, that they're really working well on some important drug discovery tasks, and really want to get that across with a lot of data. Data is the truth. And then I'll finish up with talking about where we're headed. Okay, so just a little bit about Deep Genomics. So AI, of course, and machine learning are central to Deep Genomics. We've raised over 240 million. We have about 100 million in cash in the bank right now, a few years of runway, so lots of time to get this working properly. This is kind of a map of what we're doing. So right now we're co-developing drugs with partners and building AI models with partners. One reason we're focusing on partners right now is to bring in non-dilutive funding. Just because of the markets, given where they are, that's kind of a good thing. In the near term, wholly owned drug programs. So I'll talk a little bit about that, what we have going on there. But if you've spoken or worked with any of these drug companies, there's definitely a tension between the AI platform and what's going on in the clinic. In the clinic, you spend tens of millions of dollars quickly and with risk, whereas with the platform, it's more of a continuous improvement kind of activity requiring less amounts of money. So there's tension between those two, and you'll see companies like in vitro just announced today or yesterday that they are laying off 22% of their workforce, and you can bet that that's going to be impacting the AI part of the company. Well, we know it is, because there are people from the AI team that are on the market now. So there is that tension, and our strategy of the now and the near term is meant to kind of deal with that tension. Value inflection, that's sort of the big value inflection for the company. And our goal here is major technology builds with pharma. Now, a lot of companies will talk about the asset, the drug program. Of course, we want to make drugs for patients. That's our goal. But there's a lot that'll happen between the AI coming up with good molecules and getting approval and registration of a good drug program that has not much to do with AI. And so for us, what we're thinking about is connecting the dots between the value that the platform generates in terms of predictions and what basically the entire industry would see as being hugely valuable. And I'll talk more about that in just a moment. So picture of the team. I'm going to talk about a lot of stuff today. It's not my work. It's the work of amazing team members. And I'll point a couple out as we go along, but there's no way I can really give proper credit to everyone. We just spend all of our time going over dozens and dozens of names. But here are just a few examples of people in our machine learning group. Albie's the head of the machine learning group. Bhargav is here today, right over there. We'll talk about a little bit about some of his work. And this is really where we're focusing our hiring right now, is expanding the machine learning team. Okay. So I wanted to give you a sense of what's going on at Deep Genomics in terms of the team, because that is crucial. So we have machine learning, which is focused on machine learning specialists and machine learning scientists. We also have machine learning engineers. That's in a different slice of this pie here, but we have 7 machine learning specialists and scientists, 8 supporting engineers. So those are everything from front end, back end, and also, as I said, the machine learning engineers. We have 7 people working on targets and genetics. So those would be people doing genome-wide association studies and so on. We have an automated lab with robots and so on, 10 people there. And then our drug discovery team is 9. And certain pieces of these slices are growing. As I said, the machine learning slice is growing rapidly right now. Now, I will say, and we'll see this thread throughout the talk, that I think Deep Genomics is unique. There are a lot of sort of drug discovery startups popping up where it's like an LLM for DNA or something like that, where these other pieces are not present. So if you want to have impact on drug discovery, you do need to have experts on chemistry. You do need to have actual biologists doing the wet lab experiments. So I just want to point out that this is something that I think is really valuable at Deep Genomics, that we have these different pieces of the pie. Now, the other thing I'll accentuate, and this is a thread throughout the talk as well, is that discovering truly causal relationships in complex biological systems is what we're good at. And that's what you need for drug discovery, because drugs are causal, right? That's how they work. They're causal, not correlative. And so we'll show examples where we've used machine learning systems with zero-shot learning to predict causal mechanisms that we then validate in the lab. And we've got hundreds and hundreds of such examples. And so our systems really do work that way. They're not just correlative. And then in terms of joining the team, I think that's sort of how to think about this. If you're thinking about joining Deep Genomics, not only are you going to be building these models that can actually develop drugs, advanced drugs, that are addressing the needs of patients, there's a close connection between our machine learning scientists and them working on molecules that are actually going to go into patients. And then the support of all these other team members to make that happen. As I said, everything from the machine learning engineers all the way through to the drug discoverers and chemists. So collaboration is key. I'll also highlight a couple examples of what we call lab in the loop. Other people call it that as well, which is, from the machine learning perspective, you get your data, you train your models, you make predictions, you obtain more data, you use that data to validate your models, and then retrain the model and iterate. And we all talk about that being a wonderful thing, but we all know that that's also hard to make happen. It's been difficult to actually get it to work properly at Deep Genomics, but we have really good examples now where we have it up and running with cycle times ranging from weeks to months. So I'll talk a little bit about that as well. Okay, so let's get into the AI. Now, just to give people context in terms of what's going on in the AI drug discovery community, so far it has not worked out that well. There are really 0 programs that are in the clinic that would not have been in the clinic without machine learning and AI. And I would say that's because we've tended to focus on narrow AI. And so a lot of companies have built machine learning systems that'll focus on small molecule screening, right? Or Deep Genomics would have a machine learning system that's predicting splicing, or a machine learning system that's focused on image analysis, okay? And so narrow AI is what I call that. But You might not be surprised to hear this. Foundation models are going to be hugely helpful in overcoming these limitations and these previous failures. Okay, so just to take you through the picture for the way I think about this, is to make a successful drug, you've got to get a bunch of things right. I'm showing 5 of them here. You probably need 10 or 20 things to be right for a good drug. You need a variant associated with the disease, the mechanism of the biology. You need a mechanism of the therapeutic. You also need the effect of a molecule, potential molecule, in vitro, and then also in vivo, and so on. Now, traditionally in drug discovery, you have a set of scientists that basically come up with hypotheses at each of these stages, and then go and test their hypothesis in the lab. So first of all, it's hypothesis driven. The predictions come from the brain of the human, but then it's hypothesis driven, and then testing in the lab, and then you do it step by step. Now, what we've all been trying to do for the last 10 years is build narrow AI models. So AI model to do this, another AI model to do that, or machine learning model to do this, to do that. And the problem with this is, first of all, some companies just do one thing. And so then the other 10 things you've got to get right to have a successful drug, they're at risk of failing on. So they might have a molecule that works in terms of binding, but also has a lot of toxicity. Or it's just a bad target because they didn't do the target discovery themselves. So there's a lot of reasons why that narrow AI system may be limited. Or you can do what we've tried to do at Deep Genomics, which was build a machine learning system for each of these steps. Ah, thank you. Yeah. Yeah, I don't know what a target is. Okay, so target, seriously. So target is one of these terms you hear in drug discovery, and it's a very loose term, very, very loose. So some people, when they hear the word target, they mean that's a protein you want to eliminate. So you might design a small molecule to bind to that protein, to gum up the protein so that it can't have an effect. Other people might, by target, that might mean a genetic variant that's prevalent in a disease in a particular gene that you want to then correct. And you may correct the effect of that variant by repairing it specifically using, say, DNA editing, or you might repair the effect of that variant by introducing a compensatory molecule that kind of undoes the problem created by that mutation. And so, and that's actually kind of an interesting aspect of this field. The targets mean different things for different people depending on their therapeutic modality. Also, another difference in terms of targets, complex disease versus rare Mendelian recessive disorders, where in the latter case, you can figure out the target by doing segregation of the family, things like that to figure out what's going on. In the case of complex disease, usually it's many genes involved. The biology is complicated, many different pathways. In that case, you know, what is the target? The target is sort of really the nexus of all of the biology that leads to the disease, and how you might intervene in that target can be quite complex. Okay. Thanks. Any other clarifications, clarifying questions? Anything goes. Okay. All right, so Deep Genomics, we had 40 different machine learning models 3 years ago. Each of these machine learning models having its own data set that had to be carefully engineered and designed and maintained. Each of those machine learning models having a team responsible for building the model and validating it from a machine learning perspective. Each of those machine learning models requiring orthogonal experimental data to test whether it was the real deal, whether it would really work in terms of drug discovery. And then all of these models kind of needing to be used jointly to advance molecules, just like shown here. So basically it was a mess. It was not scalable. Like if we wanted to make them better, how do you make 40 different models better? It's not scalable in terms of the teams needed to do that work. Very difficult. And a lot of companies are facing it. So a lot of the AI drug discovery companies are facing that problem. So, so here's the foundation model approach. So the foundation model approach, and really we came across this about 2 years ago, of course, you know, ChatGPT and transformers and the excitement around all of that work. When that was taking off, we realized that we could build a single model, a foundation model that would, for example, know about all of RNA biology. And I'll tell you what I mean by that in just a moment, but it would kind of know about all these different aspects of RNA biology when it comes to these different kinds of models that you might build. Okay, so that means that that one foundation model takes into account synergies between these different tasks when it's being trained. And so, for example, the variant associated with the disease is, of course, related to the mechanism of the variant biology. If I can understand mechanisms of biology better, I can identify variants better. And similarly, those sorts of things will be relevant to the mechanism of a therapeutic. That is, if there's a particular mechanism that a mutation interrupts, then the therapeutic needs to reverse that mechanism. So those are just some examples, but the idea of the foundation model is that it can achieve synergies between these different tasks during training, and then also synergies between these different tasks at application time. So dependencies between these different objectives. The scalability is much easier. So instead of having 40 different models, there's one foundation model. So if you want to put in 10 times more data, it's just one data set engineering task instead of 40 different tasks. And all of that becomes easier. Yeah.
A clarification question. The data that you're talking about, are you talking about text? Are these models reading papers, or are they like reading about like DNA sequences, reading about like experiments done in the lab,
Yeah.
the outcomes of those experiments?
Thanks. Great question. Yeah, so when it comes to AI in drug discovery, there's a lot of different kinds of data you could be looking at, like you were saying, papers, or it could be biological data. And I'll talk about this in a moment. In our case, it's biological data. So it's basically genome biology data. So that's genomes, RNA-seq, protein binding data, degradome-seq data sets, all of these different kinds of data sets that kind of let you peer into the genome biology and measure what's going on. And I'll give you some concrete examples. However, I think what I've talked about here would apply for other areas as well. And so if you're looking at a clinical, even something like clinical trial, running clinical trials, things like how you run your clinical trial will depend on the biomarker that you're using. So the relationship between, for example, the biomarker and how reflective what's going to happen in the clinic it is, which patients it will work for. So maybe the biomarker will work really well for a subset of patients, not so well for another set of patients. And so even when you're kind of looking more at the clinical setting, these kinds of ways of thinking about the problems persist. I thought I saw another hand. Okay. All right, so let's take a look at this. What do I mean by this data? All right, so Deep Genomics is at the intersection of two major fields, RNA therapeutics and causal foundation models. And I just thought I'd throw up here a bunch of prizes that have been won recently, including everything from deep learning, as everybody knows about. But also I wanted to put up there mRNA vaccines, Nobel Prize in 2023, and then microRNAs, Nobel Prize in 2024. And so, yes, deep learning and foundation models are hot. Just wanted to point out that RNA therapeutics are also on the rise. And this cartoon here, I think, kind of captures it. Yeah. So the new view here is, yes, life is a seething mass of RNA that sometimes uses DNA to take notes. And what about proteins? Errands for the RNA. And so really RNA, for example, DNA does not do anything that's tissue specific. The DNA looks the same in a liver cell and a brain cell. For most diseases, the DNA mostly is the same. For cancer, of course, a lot of stuff happens to the DNA, but for many diseases, the DNA looks the same in all the different tissues and so on. Looks the same in the disease state and the normal state, except for maybe one particular nucleotide where there's a mutation, just one. But RNA is different. RNA varies across different cells, cell types, across different tissues, hugely varies across different cell states, like disease versus non-disease. Yeah, and the proteins do the errands. All right, so what I wanted to come back to is we talked about transformers and attention. This is kind of a tongue-in-cheek remark. Attention is not all you need. You need causal models of biology. I'm not saying attention is not necessary. Of course, it's hugely helpful or something like it. But the point here is you need causal models of biology. And so to get this home, a recent paper published by Gonzalez et al. Looked at the problem of classifying mutations associated with complex traits. And what you're seeing here on the x-axis is the area under the precision recall curve for classifying those variants. And at the bottom, what you're seeing is the DNA LLM models. And so hyena DNA, for example, that some of you I'm sure are familiar with, but more recently EVO2, and there's the EVO2 model with 40 billion parameters that was released just a few months ago. And then at the top, what you see is models that are built to reflect causal biology. These are the kinds of ones we're building at Deep Genomics. And you can see that the area under the precision recall curve is 2 to 2.5 times higher when using those kinds of models. Yeah. Oh,
What
yeah.
do you need this area to be in order
Great
for
question.
these models to be
Yeah.
useful?
So what does this even mean in terms of drug discovery, right? Like what would be meaningful? Depends on what you're trying to do. If you're trying to find a drug target that is a mutation in a gene that you might be able to drug for that complex trait, then the area under the precision recall curve gives you a sense of how much searching you're going to need to do within your predicted candidates before you find one that's bona fide. That's the real deal. And so that's not an answer to the question. It's basically to say that area under the precision recall curve that's 2 to 2.5 times higher means you'll do 2.5 times less work to find that target. Of course, what I just said completely depends on other sources of information. You might have a research paper on a particular target that does a deep dive, which would give you orthogonal evidence. But the point here is just that that's what adding in causal biology gives you. LLMs for DNA are not meant to be learning causal biology. They're meant to learn whatever they can just by looking at the DNA. And we'll talk more about that. Also, single cell data. There's a recent paper just a couple of weeks ago on zero-shot evaluation of single cell models like SCGPT and UCE, finding that they don't really work that well in many tasks. I won't elaborate. We can check out that publication if you're interested. All right, so here's this approach that we take. So this underlies everything we do. So our foundation models are trained on data that reflects the causal relationships of genome biology. So you're seeing here the central dogma. We go from genomes, a genomic sequence at the top, that gets transcribed into a pre-mRNA, a nucleotide sequence. That gets processed into mRNA, mature RNA, which then gets translated into protein. And you're seeing all these little arrows and blobs. Those are all sorts of different processes that are involved in these steps. And there are many other processes, including ones that are known and unknown, that are not shown here. What we do is we collect data, including perturbation data, for basically you can think of it as inputs and outputs of these processes, and then we build foundation models for those processes. Okay. And everywhere you see a little box with dots in it, that's a data set that we have. And there are more. I'm not showing all of them. But for example, we have chromatin-seq to model chromatin effects. We have genome sequences, of course. We have RNA-seq to measure RNA expression. Protein binding data sets to tell us where proteins are binding and the effects they're having. We have data sets that allow us to look at RNA editing, so ADAR. We have data sets that allow us to look at microRNA binding, all of these different processes. Now, a lot of those terms I just mentioned, you might not know, but you don't need to. Basically, this is the central dogma of biology that captures the causal relationships that we know about in genome biology. And there are lots of data sets that allow us to peer in and measure what's going on. And so that's the key, is if you know that A causes B, then you can gather data for A and B and build a machine learning predictor of B given A. And that's the key here, right, is knowing about these dependencies that scientists have discovered painstakingly through experimentation.
How much of this is automated for collecting the data?
We just push a button and then everything happens. All the data sets are collected. No, there's a wide range of answers to that question. It's a great question. So at Deep Genomics, we have a large team of bioinformaticians that work closely with the machine learning scientists and machine learning specialists to gather the data sets, to change the formats, to convert them to different formats. Which reference genome are you using? The data set needs to be aligned with the right reference genome and so on. And so there's a lot of support staff at Deep Genomics that do all that work to formulate the data sets in collaboration with the machine learning specialists and the biologists. Some of these are more automated than others because we've been doing them a lot. Like RNA-seq now is pretty automated. Other ones are a bit more bespoke, like the data set for ADAR, RNA editing. One person put that data set together and built the model. So it's kind of a range of answers to the question. Great. Other questions about this approach. Yeah.
So the input to the foundation models are the DNA sequences, or these are the output that foundation model are trained to predict?
You mean in this diagram here?
Right.
So everywhere, for every process, like for example, splicing here, right, which basically removes these introns from the mRNA sequence to obtain this sequence here. So that's what splicing is doing. We would have data sets that are measuring the abundance of the RNA sequence. We'd also have data sets that are measuring the binding of proteins, right? Because binding of proteins can modulate splicing. And then we'd also have measurements of spliced mRNA. And so given those data sets, we can then build a model given the pre-mRNA and given the proteins and the proteins binding, we can predict the spliced RNA sequence.
I see. So the input is also the binding, not just the sequence alone, right? Because a lot of foundation models are trained to predict the sequence nucleotide.
Or it can be the that can be the output of another model, because another model will be predicting binding given the protein identity and the sequence. So we're kind of stacking them all together as well.
Because I thought you mentioned you have one single foundation model, right? Instead of 5 models. Right?
Yes.
So
yes. Good question. Okay. Yeah. So as soon as you have a foundation model that does everything, the next thing you do is you need to have more everythings. It's like galaxy. You discover the galaxy. Wow, that's the universe. And no, there's billions of galaxies, and then maybe there's billions of universes. So same kind of thing. As soon as you get good at doing something with a foundation model, you then want to expand your space. And the question is, do you just add the data and try to cram it all together into one giant foundation model that does everything? That's just a question of scientifically what you can do step by step. So the first thing you do is build a foundation model for that other area, and then you'd seek to combine them. So we'd have a foundation model for DNA, we'd have a foundation model of RNA given DNA, and then over time we would seek to build one foundation model of both. Yeah, Joshua?
So, sorry. So the perturbations here are really quite important because just matching input-output pairs, even though the output comes from simulating under a causal model, wouldn't guarantee that you would then mimic a causal model. Like the actually being able to perturb the model and, and get outputs under a variety of perturbations is quite important here. So in this system that you've drawn, like what all can you actually perturb? Because my understanding is that some of these steps are actually pretty hard to go in and perturb.
Yeah. Yeah. So nature provides a lot of perturbation. And so, for example, if you're interested in if you're interested in building a model that tells you how genetic sequence will determine splicing, you can just look at genetic sequences across the entire genome, of which there are like 200,000 examples of those sequences. And then you can build a model whose goal it is to predict splicing from the sequence in a way that is invariant across those examples. So instead of like a very traditional biology focus would be you only look at that one instance, and then you want to know what's going on there. So you want to perturb that one instance. But a machine learning, another perspective is you say that all splicing will occur using similar rules across the genome. And so by looking at examples of differences across the genome, we can learn how those differences lead to change in splicing. Another example would be patient mutations, like just natural genetic variation between us will lead to changes, so natural variation, not maybe disease specific, but natural variation between our genetics will lead to changes in things like RNA expression levels. And so that is a natural perturbation. Yeah. Now those natural perturbations are biased, of course, in many different ways, but any perturbation experiment. Pardon me?
The biology invariant that you can still learn from these natural experiments.
Yeah.
So you're handcrafting the causal graph, right?
Yep.
What if the biologists are missing some pieces? Maybe there's some, edges that are missing, maybe some other parts of the state of the cell or the body isn't being taken account in those tables.
Yeah, that's a great point. So definitely there's a lot of stuff that's missing. And one thing that's true about biology, there's always exceptions to these supposed rules, right? There's always going to be weird things going on for certain cases. And like the example I just gave of viewing all these 200,000 examples of splicing as sort of being like independent training cases to learn a model. That's not true, because things like the chromatin will change shape, and when the chromatin changes shape, that's going to en masse change splicing for all of the splicing events that are within that chunk of chromatin. And so first of all, the graph isn't correct. And then second of all, our assumptions that we have kind of independent data are also not correct. Yeah, but the good news is we can talk about it. And so we have a framework where we can say, maybe there's an arrow that's missing. Maybe we need to explore this. If somebody comes up with a new discovery, we can map it to this framework and say, let's change this framework. If we have this question about examples not being truly independent, we have a framework to talk about it. We can say, okay, the chromatin might be inducing dependencies between these examples. And so I think that's what's most important, is that we have a framework to actually address exactly those questions.
So my understanding is so it's trained to use like a labeled task, but like an LLM also trained to use like a non-supervised task. So
yeah,
like
that's a great point. Unsupervised versus supervised. Yeah, we could go on and on about that conversation. It's a really good, good discussion point. And I'll just say for now that it's kind of like, the way I view it is that we kind of go back and forth between them. Like when you have a good supervised task, maybe you've got sort of like an autoencoder type of structure or self-supervised learning structure. There's all these sort of things in between, fully unsupervised, that allow you to kind of explore the space of trying to predict labels from inputs. And then as that space gets explored more, you can start thinking about what would be a generative model or an unsupervised learning method you could imagine using. The other thing I'll point out, by the way, and you're going to see this in just a moment, that the labels here, quite often we have 3,000 times more labels than inputs. So literally for each nucleotide of DNA, we have 3,000 examples of labels being predicted. And when you have that kind of asymmetry, then there's an interesting question of, it's not the same thing as like a supervised learning task where you have a huge amount of input and just one labeled output. Joshua? I think do you want to respond to that, or? No. Oh, okay, let's go on to the other question and we'll come back to Joshua.
Yeah, I had a question about whether you were also using non-human data or only human data.
No, we use mice as well and NHPs, and cell lines, like cancer lines, that kind of thing. Yeah.
And metabolomics? Because
no, not at this point, but we're interested in that. Yeah.
So I imagine that for any of the actually observed data sets, there will be other variables in this big graph that were not measured in the experiment. And so you should be treating them as latent variables in all the computation you're doing. Are you?
Great question. Yeah. So we have done that sort of thing in the past. And
you worked on this for your PhD.
Yeah, exactly. Yeah, that's right. Yeah, it's a really good point. I would say that, yeah, with deep learning, just getting the systems up and running to train, especially when the amount of data is massive, is in itself a challenge. To now layer in trying to sort of account for hidden variables in a sort of structured way is now it adds complexity, but
the algorithms exist. We have a lot of amortized inference.
Right, we have techniques to do that. Yeah. What we try to do to try to limit the complexity of training is we try to pick chunks here where we reduce the number of hidden variables as much as possible, right? So we're sort of, instead of trying to go from A all the way to D, where there's going to be a bunch of things we haven't measured, we try to go just from A to B to minimize the number of hidden variables. But there still are hidden variables, of course. And even outside of this framework, there are obviously hidden variables we don't know about. And so there is this issue of, I used to be a big believer that in latent variable models, and that maybe you could infer causality. And now I think you just got to measure causality. Yeah, don't try to infer causality, just measure it. Could change my mind though, yeah.
I could respond to that, but I am curious about evaluation here because my understanding now is that you have some system that, given inputs A will output B in a causally consistent way because of all the perturbation data you have. But like, it's not going to surface at the end seemingly a causal graph directly or something you can directly interpret. So how do you evaluate how good the system is? Like, I think like extrapolation to novel perturbations would be the most obvious way.
Yeah, that is it. Yeah. And so it's a great question. I also, I'll say something that's sort of my personal view right now, that model interpretability is a bit of a red herring. And I know people will disagree, but I think what's more valuable from an AI system is that it makes predictions for real world causal relationships that can be tested in the lab. And the analogy I make is that we've got, say, brilliant scientists. When we want to understand whether that scientist knows what she's talking about, we don't say, I want to peer inside of your neurons and synapses and see what's going on. We say to that individual, here's a question, give me your answer. Or tell me, if I prod the system in this way, what'll happen? That's how we build confidence that that individual actually knows about the causality of the system that they say they know about. So that's my view on that, and I'll give you lots of examples of that. But I got to get going so I can show you all these. I'm going to power through. Okay, one more question. Yeah.
So I wanted to go to the question of latent variables, and if there are variables that are hidden in this graph, can't we see this framework as implicitly marginalizing them out, and do we really need to infer them?
Well, you could, except for the fact that our machine learning systems are, the way we're training our models, we don't explicitly account for that. So we're not explicitly marginalizing them out. But yes, effectively, because we're not measuring them, the data that we have has them marginalized out. Yeah, that's right. Okay, so let's, I'm going to skip a slide here just because I want to show you the data. The data is exactly going to answer some of the questions. This is a complicated diagram. It's just meant to show you the different kinds of things we're working on. So we ingest or build foundation models, and then we make them better, faster, and application specific. And so we definitely download publicly available models, and then we re-engineer them, make them task specific. All sorts of different inputs here from proprietary data sets that we've generated through to genomes and perturbation data. All sorts of different applications, so therapeutic design, prioritization of targets, novel targets, all of those things I said you've got to get right to have a good drug. And we focus on DNA and RNA biology, so that's nucleotides, essentially nucleotide sequences, protein biology, and then systems biology, which is the one that's most nebulous up there in the far right. What does that even mean? But it means interactions that you cannot account for just by looking at a single gene, single mutation kind of thing. The foundation model platform itself, as you pointed out, it's not just one model now, it's a combination of different foundation models. Is tightly linked to our wet lab, so that we have this lab in the loop capability, and I'll show you an example of that, where we can validate and also retrain the model, make them better, make the models better and better over time. Okay, so let's look at some data. So I have 3 vignettes to go over with you. And as I mentioned before, we're focused on this genome biology or central dogma structure, and so we can peer in 3 different places, look at 3 different models: big RNA, repress, and deep ADAR. All right, so big RNA, I won't dive into the details of how it was trained, but I will show you what it was trained on and what it can do, so you can get a functional understanding of the model once it's trained. Trained on over 1 trillion signals, genomes, RNA-seq data sets, protein binding data sets, other types of data sets in that picture of the central dogma. Training and validation, I'm just giving you a snapshot of the architecture. We won't dive into it in much detail here. Again, though, the different disciplines within Deep Genomics were all critical in building this model and also validating it. So asking the question of whether it can predict with zero-shot prediction a patient mutation and get the mechanism correct, for example. And then on the right, everywhere you see a Deep Genomics logo is a process for which big RNA can make a prediction. And what I actually mean by that, I'll tell you in a moment, but just to show you the idea that it can make predictions across this entire Central dogma of molecular biology. Okay, so this is sort of meant to give you a clear idea of the input and output of big RNA. It's a sequence-to-sequence model. So the input is 1 million nucleotides or less, basically a gene, an entire gene. All right. Introns, exons, 3' UTR, 5' UTR, the entire gene sequence. And then big RNA will make predictions for over 3,000 different molecular biology tracks. Okay, so the x-axis here is position within the gene, the nucleic acid position. And the y-axis on the first one is the RNA-seq expression level predicted for that nucleotide in a particular tissue. There are 50 different tissues here, so you're just seeing the track for one particular tissue. We also make predictions for riboseq, which is whether the machinery is attached to translate the RNA into protein, predictions for protein binding, predictions for microRNA binding, and so on. Now, once it's trained to do this, and this is where what I mentioned before, 3,000 different tracks as targets for that one gene. Once we've trained the model on the data that we've trained it on, we can then make zero-shot predictions for all sorts of things. We can look at disease mutations. We can introduce a disease mutation and ask, is that going to cause skipping of an exon? And so maybe this particular piece right here drops down, disappears. Is that particular mutation also going to cause disruption of protein binding? If it causes disruption of protein binding and decrease of exon inclusion, you have a mechanistic hypothesis. The mechanism of that mutation is it prevents binding of the protein, which leads to exon skipping. And so you can quite easily look at these tracks and figure out, first of all, predictions for what you can go measure in the lab, and then predictions for the mechanism by which that effect could occur. And I'll show you examples where we validated that. That's just one thing you can do. You can also simulate the effects of an oligonucleotide. So if you're wondering if an oligo could restore that exon and overcome the consequence of that mutation, you can simulate the effect of the oligo. And an oligo is basically just like a 20 nucleotide sequence that binds to the complementary sequence in the gene. So literally just one-to-one matches a particular 20 nucleotide sequence in the gene. So we can simulate the effect of that oligo by going to those inputs and just setting them all to 0. It's just ridiculously simple. We just set all the inputs to the neural network to 0 for that 20 nucleotide stretch. Remember, we're going to use one hot encoding to encode the nucleotides. So we set them all to 0, run it through big RNA, and then ask the question, is that exon restored?
So there's data for this kind of experiments?
We have lots of experimental data, yes. But what I just...
With oligos?
Yes. But we found that our zero-shot predictions work extremely well. Pretty amazing. So this is a model that's never seen oligos, and it can accurately make predictions for how an oligo will change biology. Yeah. DNA and RNA editing, same thing. You have a CRISPR system that you're going to use to make an edit. Maybe you cannot repair the mutation, but you can introduce a compensatory mutation somewhere else. You can run it through big RNA and make a prediction. If you're interested in designing an mRNA therapeutic, the 3' UTR, the end of the gene, turns out to be really crucial for getting tissue-specific and expression of an mRNA and really good expression of the mRNA. So a model that can predict how changes in that UTR can impact expression is useful, and that's what big RNA does. Okay, let me show you the data. All right, so quickly here, I'm going to show you some examples, but just to give you an overall sense of what's going on here. This is like a machine learning metric example. So I'm going to show you machine learning metrics and drug discovery metrics and examples. So as machine learning scientists, we kind of like to say, what's the area under the precision recurve, call curve? And as a drug discovery, we want to know, okay, but did we find a drug? So those are the two things we'll talk about. So here are machine learning metrics. We looked at 5 different tasks: protein, RNA binding, exon skipping mutations. So those are patient mutations that cause exon skipping. Splice switching oligos, I just gave that example. An oligo that binds then causes splicing to change, and a couple others. And for each of these tasks, of course, there's many different ways you can measure performance. There's false positive, false negative trade-offs and specificity and all of that stuff, correlation, Pearson correlation. But for each of these tasks, we just picked one that we thought made sense for that task. And then we compared big RNA with other state-of-the-art methods that are out there. So DeepRipe is another method that's out there on predicting protein RNA binding. Big RNA achieved an accuracy of 91%. DeepRipe achieved 11%. And this is an accuracy that, of course, as you might guess, that we had to set some threshold on false positives or something. If we'd set it really low, then like DeepRipe would have been 0%. So we set it to some level that made sense. And you can see for the other tasks, so Big RNA versus Splice AI or Informer or Alpha Missense from Calico and DeepMind. Saluki is another method. Target Scan is a microRNA binding predictor developed at MIT. And so you can see across the board, it works really well. I'm not going to spend much time on this because I want to kind of dive into more of the causality stuff. So here's one example. We have, as I said, we have hundreds of examples like this, but here's just one example. I want to take you through it so you can see what's going on with the model. So the model input shown on the left is a healthy sequence using one hot encoding for the nucleotides. And for this particular gene, this is ATP7B exon 6, which is involved in Wilson disease. If you look at exon 6, Big RNA predicts that that exon is expressed. So you just see a big bump there of expression on exon 6. When we run the disease mutation through Big RNA, that's shown in red down there in the second sequence. So we switched an A to a G. Big RNA predicts that the exon is no longer included. All right, so the expression level drops down. All right. Thirdly, we can scan a whole bunch of oligos across the sequence. And you're just seeing one short sequence. We scanned it across the entire gene and looked at lots of different locations. We can simulate the effect of the oligos I mentioned before. And if we place an oligo as shown here and run it through Big RNA predicts that the expression level is recovered. It's even a bit higher than the naturally occurring expression level for that exon. Okay, so those are the predictions from Big RNA. We then went to the wet lab and validated. So this is a prospective validation of the prediction. I won't spend a lot of time going through the experimental procedure, but basically we CRISPRed that mutation into a cell line. We measured wild type and homozygotes, and also the ATP7B variant on the far right. And you can see that the prediction was correct, that mutation does lead to a drop in inclusion of the exon. Regarding the oligonucleotides, we actually tested hundreds of oligonucleotides because we wanted to know not just whether we happened to by chance get it correct with this one, but we also wanted to have positive and negative data. So we measured hundreds of oligonucleotides. That's shown in the red line there. So the x-axis is the location of the nucleotide with respect to the exon, and the y-axis is the fold change, how much we boosted the expression of the exon. And then in gray is predicted. And so you can see predicted and measured agree pretty well, a correlation of 0.91. Okay, so we figured the mechanism of the variant out, and we also predicted the effects of these oligos.
So for this in silico mutagenesis experiments, if you do, say, discontiguous oligo, so that's going to be combinatorial, right? So because you have 1 million sequence long, if you test all the combinations, it's still going to be very expensive.
Yeah.
I wonder whether you have done something like a gradient aggregation, where you can, aggregate the gradients at nucleotide level, and then which, suggest what nucleotide to change.
Yeah, these are great ideas. So we've tried a lot of different things out. Combinations is one thing we can try out. I'm going to show you a little bit of data later, I think, for mRNA, where we looked at, and this is work that Bhargav did, where we looked at the 3' UTR and added in mutations to try to create the best, most stable version of that mRNA, and then looked at how the aggregate effect of all those different mutations would benefit the stability of the mRNA. Yeah, and so, but you got the right idea. Anything you can come up with in terms of what you'd like to explore, you can just run it through the model and see what happens. And then do the experiments to validate it, of course. All right, we're going to take another look at another thing. I was just talking about this, the 3' UTR. So this is the work that Bhargav did, repress. And so this one's interesting. It was trained on internally generated data for microRNA binding and degradation of mRNA. That's what leads to lower levels of mRNA in the cell. And so our wet lab and in collaboration with the CRO generated large amounts of data. I'll talk about those in just a moment. And then we built this model here called repress, capturing the idea of repression of the mRNA. And it takes as input a 12.5 kilobase context, as shown at the top. I won't spend a lot of time again on the architecture, but I think a lot of people in the audience will kind of recognize the different architectures here. This is like a conv-next type architecture. And it predicts two different types of outputs, as shown down here. One of them is the microRNA binding probability on the left, and the other one is the degradation probability shown on the right. And so that's just saying that for a certain sequence, it might get degraded more frequently than another sequence. That's the information that's captured there. Again, these are sequence-to-sequence models. Now, okay, why is this important? Why is this useful? As I said before, we could do things like play with the input on the mRNA and mutate it until we can find one that is degraded less and has fewer microRNA binding events, and therefore will be more stable. That's the kind of thing we could use this model for. All right, so ML metrics. Again, we're going to do ML, drug discovery, ML, drug discovery. So I don't have time to go into these in detail, but we tested repress using 8 different tasks. Those are shown on the x-axis down below. So those are 8 completely different tasks in drug discovery that involve microRNA prediction. So they're all related to microRNA prediction. These are different data sets from different groups, the different tasks. And then we compared repress with 7 other models, and those are shown on the x-axis. And for each model and way of testing the model, dark red means that model performed the best, whereas white means it didn't perform well at all, except for the pure white. Those are the pure white blocks is where the model was trained on the corresponding data set. And so we did not evaluate it on that data set. That would have been kind of cheating. Even though we always do cross-validation, even just using the same kind of data is, when it comes to biology, is sort of cheating. Okay, and you can see that repress performs better than the other techniques across the board. We also looked at different architectures. So here on the right, you're seeing either area under the precision recall curve or Spearman correlation for predictions from repress. Repress is shown on the left. That's a 33 million parameter model. But we also looked at dilated convolutional neural nets. We looked at Mamba, we looked at transformers, and vanilla convolutional neural nets. And you can see that this conv-next type architecture works substantially better than the others for this task, of course. All right, now let's look at the drug discovery aspect of things. So here there's 3 subvignets for this one. So we're going to look at antisense oligonucleotides, siRNA, and also mRNA therapeutics. So the one on the left, we took data for about 2,000 different ASOs that were designed for a gene. We scored them using repress. Antisense oligonucleotide. All right, so that's this thing I mentioned that just Watson-Crick matches up with your target gene. So nucleotide by nucleotide matches up and then has some effect on that target gene. So data for 2,000 of those antisense oligos across this gene of interest, UTRN. We scored them using repress. And then what we do is we look at the oligos that were taken forward by others as therapeutics and ask the question, would repress have discovered those? This is a retrospective analysis for whether we could have used it to identify the oligo that was taken forward. We have good statistics. We have end up with a good correlation between the predictions and the measured data, but I'm just showing you one example here. In this example, you can see that here's the end of the body of the gene. Here's the 3' UTR, and these are the predictions for where the microRNA will bind. If we zoom in this region here, here's the prediction from repress for where the microRNA will bind. And here's an ASO that was taken forward as a lead compound by another group, and it blocks that microRNA. That was also the top scoring region in that UTR, according to repress. So repress recovered that compound. Another one here, let's look at siRNA. So that's Alnylam is an siRNA company. These are oligos that bind to a target gene and then repress it, turn it off. And similar approach though, we looked at data for 1,000 siRNAs, scored them using repress, and then analyzed the corresponding siRNAs. In this case, we looked at a different measure. We decided to look at how many siRNAs you would need to screen to find the top, say 50% of the actually good siRNAs. And so on the x-axis, number of siRNAs tested using the different prioritization of those siRNAs. using repress or using the industry standard method shown in orange. On the y-axis is the fraction of good hits identified. And so, for example, if you look at 50%, so we want to find 50% of the good hits, we see that repress can do that by screening 5 times fewer siRNAs. So a huge increase in throughput. And the last example here is on mRNA. So think of Moderna, for example, where you're looking again at this 3' UTR. You're trying to modify it to make it as stable as possible. And that's what you're seeing with that little movie. You're seeing different mutations being added in iteratively. And as those mutations are added in, repress is being used to score them and then choose which sequences to proceed with in terms of adding more and more mutations. So that's work underway as well in terms of obtaining additional experimental data, but we already have some data in. On the x-axis is the machine learning design score from repress. The y-axis is the actual mRNA level, so how stable it is. And we get a correlation, as you can see here. And particularly the ones that do have the highest score tend to have the lower mRNA levels. The ones that have scored near 0 have higher mRNA levels. Now, how all this translates to actual drug discovery is in the works right now. It's something we're working on today. Okay, and the last one I'll talk about is Deep ADAR. So this is a model of RNA editing. What is RNA editing? So RNA editing is a naturally occurring process that's happening in all of your cells right now, where A's are converted to G's, and that'll happen if your RNA forms the stem loop structure shown on the left. Okay, so your genes are being transcribed into RNA, your RNA folds on itself and sticks to itself. If it forms a stem loop structure and there's an A present, this ADAR machinery comes along and edits the A to a G. Ubiquitously across your genome and on all of your cells, that's what's going on. And so the idea for a therapy is instead of having this naturally occurring secondary structure, just induce that structure by putting a short RNA molecule in there, say a 40 nucleotide RNA molecule in your cell that binds and forms a similar kind of structure. That'll then recruit the ADAR machinery, convert an A to a G. Good question. That is one of the questions. Hopefully it's local, right? So that is a good point. You want to design that molecule so it'll be as specific as possible. But that is a challenge. Perfect challenge for computation. So there's 3 ways you could design a therapy that we can think of. One of them is actually a disease involved an A that got converted to a G. So you just want to go in there and convert the G back to an A. That's the simplest one. The second one is inserting an edit in a regulatory element of gene expression so as to boost the amount of protein, for example. It could be, for example, we're just talking about the 3' UTR where you're trying to increase stability of the mRNA. If changing an A to a G could substantially increase the stability. That could be a good target. And then the third one is an edit within an exon that'll change the function of the protein that's produced. So you change the exon, you change the amino acid, change the function of the protein. That one's much harder. People are working on it, not a lot of progress yet, but we're hopeful. All right, so where are we on this in terms of the ML metric? So we take 1 million naturally occurring edit sites, we train Deep ADAR using, as you might guess, the input sequence, but also RNA structure features. Those are obviously relevant to this task. The targets in this task are just the scalars predicting editing efficiency. Once trained, we can, of course, validate it using the natural editing, just using RNA-seq data and natural editing data. But we can also predict the effects of a guide RNA-induced editing event. That's where we synthetically put in that RNA sequence to induce an A to G edit. We've done that and obtained a bunch of data, asked the question whether we can predict editing, and achieved here a correlation of 0.76, again on zero-shot prediction of this process. In the terms of the drug discovery part of this, we're aiming to have 2 to 3 RNA editing programs in mice by the end of the year. So that's where we are. The lab in the loop idea has been really critical here. So in the upper left, we design these guide RNAs using our system Deep ADAR. We then manufacture and screen them in a cell-based assay. Obviously, we sequence and analyze the data. We then retrain Deep ADAR and iterate. And our current cycle here is about 2 months. So every 2 months, we can obtain a new data set, retrain them all, deploy it, design new guide RNAs. Now, out of the sequence and analyze step comes molecules that we're advancing into cell-based functional screening. So are we seeing the neurons being fixed correctly? Are we seeing metabolic levels being adjusted correctly in our cell system? And then we'll advance them to in vivo testing in the very first part of 2026. All right, so to wrap up, I just want to say a few things about where we're headed in terms of AI across the company. And the first thing I'll talk about is accounting for disease context. And so we talked about big RNA, and the version of big RNA I spoke about was just trained on naturally occurring data. It wasn't disease data, just naturally occurring data. It's cool, though, that it could generalize to disease states, right? A mutation for a given disease. But sometimes a disease state changes the cell state, like changes the proteins expressed in the cell, changes or more complex systems, such as even tissue-to-tissue type of systems. So a disease state can be quite complicated and have a big impact on how the gene regulation will work. So what we did here is we took big RNA, we generated or obtained data for TDP-43 knockdown, which is a protein that is sequestered in ALS, so it's not available in ALS patients. And then ask the question, okay, given that TDP-43 is not available, can we train a version of big RNA that takes that into account and then can make more accurate predictions for targets and for molecules? And so we had this workflow here that was designed to obtain this model, big RNA ALS, a disease customized model. So we then used big RNA ALS to analyze mutations associated with ALS and score them and rank them. And that's what's shown on the far right. And big RNA ALS more accurately identifies validated variants compared to the standard big RNA model. And so, for example, the mutation here that Lilly is taking forward is Curalis. That's the RS number for the variant. With big RNA, it was ranked 33 in terms of its predicted effect. With big RNA ALS, it moved up to rank 4. So it has a much larger effect. And as you might guess now, we have greater confidence that these three up here, which are novel, could be valuable in terms of therapeutics. And so we are pursuing those right now. So big jump in terms of accuracy for the disease-specific model. The second thing I'll comment on is scaling laws. I think a lot of people here know about the importance of scaling laws. I alluded to it at the beginning that you would like to have a system where you can put in more data and everything gets better, or you can put in more compute and everything gets better. And this is some data that Bhargav obtained on Repress, where we are seeing that, and it's actually quite impressive in terms of what the curves look like. So the one on the left is the number of proprietary data sets that we've generated. So we've used 18 public data sets, but on the far left, we're using 0 proprietary data sets from Deep Genomics, and then on the right, there's 20 proprietary data sets, so a total of 38 data sets. And you can see the accuracy getting better and better and better, from 20% to 60%, and it's still going up. So we'll generate more data and get more accurate. On the right, you're seeing the effect of scaling up the sizes of these models. So we've looked at a lot of different model sizes, increased the scale of the model, both in terms of the architecture, also just in terms of the number of parameters. Here you're just seeing number of parameters, and again, you can see performance is going up and up and up. All right, and then the last comment I'll make, causal biology is what you need. So after all of this was done, we just said, what happens if you just go to ChatGPT and say, for this patient mutation, what's the mechanism, right? Just like the crudest thing you could imagine doing. We actually did things that were a lot more sophisticated than that. We wanted to give ChatGPT the benefit of the giving it additional information, pointing it to literature, stuff like that. So we built a system that could do that, and then we looked at 4 different LLMs, they're shown on the x-axis here. And then we compared the LLM with the LLM if we also gave it predictions from Deep Genomics, right? And asked, does Deep Genomics predictions actually improve things? And they do. So first of all, it was actually surprising. The LLMs do a pretty good job. And so on the y-axis here, we're looking at the number of correct interpretations of patient mutations. And so you can see the LLMs kind of get 4 or 5 correct of those patient mutations. When we also provide DG's predictions, that jumps up. It roughly doubles in the case of Llama. Gemma, it's even more than doubles. When we average, so we also said, okay, what happens if you average the LLMs? Like really give them the benefit of the doubt and take the average of all the different LLMs and all the different, for each LLM, you can run it many times. So we just aggregated information as best we could across LLMs. And when you do that, the Deep Genomics information boosts performance by 70%. All right, so good thing. Wow. Imagine if at the end of this, ChatGPT was as good as Deep Genomics. That would have been disappointing. Okay. All right, thanks. And I'll end it there and take questions. Yeah.
All right, we have a bit of time for additional questions. Yeah.
Okay, hi. Yeah, sorry, I have so many questions, but I'll try my best to stick to one. Actually, this slide is super interesting because in all cases, Deep Genomics predictions actually help. But if you compare the baselines for all the LLMs, they're kind of similar, but then for Llama, let's say, adding the predictions has a kind of exaggerated contribution, right? So for some LLMs have differential kind of boost in performance by adding the predictions.
Yeah.
Was that just something surprising, or?
All of this is surprising. The whole thing is surprising. The fact that we can predict mutations with zero-shot learning is surprising. Yeah, I think it's interesting. You know, some of these LMs seem to be able to take our data into account more effectively than others. Yeah, and I don't really have an answer for why. Chris Cole, who did the research on this, he might have some insights, but yeah, I'm not sure at this point.
So my original question actually is about ensembling, and this slide also brought up this question I had already. So I think in big RNA, you had a part that explains that you're assembling 7 variants of big RNA, but you could ensemble different deep genomic models, right? If you wanted to create a meta model out of that, right? So is that something you thought of? And if that's something interesting, would you also want to try to ensemble some of these other models that you yourself have shown work decently well, such as Informer and Borzoi?
Yeah, great question. Yes, and you know, we do have ensembling going on at different levels, as you might guess, like as you pointed out with big RNA. In terms of ensembling models made by different teams, like you mentioned, Informer versus big RNA and Borzoi and all of these different models, or within Deep Genomics, like repress big RNA could be ensemble to make a prediction. That's a great idea. Yeah, good idea. I think we've done a little bit of it, but maybe we could do a lot more of that actually. Yeah.
Okay, sorry for double dipping, by the way.
Hi, thanks for the talk, Brendan. I had a little question about the problem of overfitting in foundational models. So I don't know if you've heard of that story, but at the beginning of the year, some researchers tried to evaluate a lot of state-of-the-art language models on the 2025 math Olympiads.
Mm-hmm.
And all of them performed really poorly, showing that there might be some sort of catastrophic data leakage from evaluation to training on foundational models. So I was wondering, how do you try to prevent this sort of leakage, especially when your data comes from biology and is really hardly processable by humans?
Yeah, that's a really good point. Like, first of all, the fact that these models supposedly don't overfit has been shocking to me ever since, you know, that came out. so why aren't they overfitting? And that, you know, there's a lot of thinking about why they're not, you know, different perspectives on why they're not. And I do remember in the early days, my main concern was that these models were just memorizing examples from the training set that then turned out to be, you know, turned out to be impressive when you looked at predictions. I mean, I think we can all agree that they're not overfitting to that extreme. It's not just memorization. But what is it? Yeah, I don't know. It's hard for me to tell. I'm not running the experiments myself, so I can't check. Bhargav, are the models overfitting? No. Yeah. Well, we do. Okay, so they probably are overfitting, but
the question is, how can you make sure?
Yes. So as I said, a lot of the experiments we do, as Bhargav mentioned, the prospective analysis, we're like, for example, that Wilson disease case where I showed that it predicted the mutation and the effect of the oligo, we actually left that gene out during training. So we retrained the entire model without that gene present, just to make sure it was not learning something about that gene. Also, when researchers are building machine learning systems that are trained on publications and papers and publicly available data, then I think it's really hard to know whether or not they've actually seen examples of what you're trying to make a prediction on. For us, because we're really focused on genome biology and we're not looking at the literature at all. Like for example, big RNA has never seen any papers on Wilson disease at all. It's only seeing genomics data sets. So that makes it easier for us to know that we're not cheating in that way. And so all of the examples I showed and all the metrics we showed, like the bar chart, we held out the examples for training. Having said all of that, biology, how does crossover works? It takes chunks from here and there and puts them together and stitches them together to make the zygote. So, and so it's not bulletproof that that's not happening to some degree. Yeah. So it's a great question that we struggle with all the time, but it's a really important one.
Yes. So about your big RNA and repress, you showed examples where the effect were in cis, right? So for the gene, you have a mutation, an edit, or an oligo that is, and then you predict for that specific gene. Is your model able to predict for trans effects, like across the genome?
Yeah. So the way we take into account trans effects would be, for example, the protein binding data, where we're measuring protein binding events, which is whether or not that protein binds is a trans effect. But we don't have a so those proteins are identified as unique entities within the model. They're not. There's no model of proteins that would enable it to look at an arbitrary protein and determine whether it will bind. It just knows that this particular protein is likely to bind here, 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 and here, and it can predict how a mutation might disrupt that binding event for that protein. Does that make sense? So it's sort of protein by identity rather than the model knowing something about how protein structure and how all that works.
Yeah. And so could you use this in order to predict off-target effects?
Yes,
in a sense?
definitely.
Okay.
Yeah, that's right. It's another great example of something you think you get it. And now you can imagine all the different things you could do with the model. Yeah, and then you try those things out. Sometimes it's amazing, zero-shot learning, and it works really well. Sometimes it doesn't, and then you can think about whether you need to fine-tune the model so it can work better. But that's exactly the way to.
But then again, this is very difficult to prove or to kind of validate in the lab. But you have a system where you could then go back to the lab and then verify if those off-target effects are actually happening.
Yeah. We have lots of examples of that. Because sometimes we do protein knockdown experiments because we want to put the cell in a certain state so that we can measure things that would be hard to measure if the protein were expressed, and that does work. Yeah.
Thanks.
So you mentioned the best way to do causal biology is to measure the actual mutation and then the resulting phenotype. But there are a lot of biobank data, like UK Biobank, where you have 500,000 individuals, where you have naturally occurring single nucleotide polymorphisms, and there are also thousands of phenotypes being measured through electronic health record. So I wonder, is there any plan to train a foundation model on that type of data, where you take a genotype and then you predict phenotype? So then, the phenotypes observed directly from the genotype, and then that is more accurate label, so to speak, right? Obviously, there's a challenge in training such a model because you have to account for millions of SNPs mutation, which is not very scalable for the current model.
Yeah. No, that's a good question. We sort of I would put that like in the systems biology bucket. And I think we're just getting going on that. That's something we are interested in doing. There's sort of different directions you can go when it comes to phenotypes. And we're sort of trying to figure out how to also bucket them in a way that's specific to diseases that we're interested in too. But yeah, that's a really good point. Now, there might be some other questions, but I think at this time I'm supposed to hand it over to Joshua because we've got a little bit of a special event happening. Oh. or not, I don't know. Oh.
Yeah, so we have a bit of an announcement. So, I think you've all realized this, but T-Talks are a tradition here at Mila, and they're a tradition in other groups, and they're, slightly maybe different ways of doing it. But I remember certainly in Toronto, we had the T-Talks. And for me as a grad student, it was an amazing time to, of course, learn about the science and get a lot more breadth, but also build a community, right? It was this 1 hour a week where you'd go to and everyone would be there. And of course, after discussions would ensue. And so I very much enjoyed these e-talks. So today is the last T-Talk that RIM is organizing at Mila.
Did you know?
RIM has been organizing the T-Talks for the last 6 years. So, think of a place where we were maybe low hundreds to now where we're over 1,500. Remember maybe times during COVID. I certainly remember my experience of like logging in and there was this nice calming music, which I always found reassuring in these times and calming. And so I think on behalf of everyone, and I'll let Yoshua speak, of course, I just wanted to give you a big thanks. It's been, the T-Talks have been the core of our scientific animation at Mila. And thank you so much, RIM, for organizing them. If you've ever organized anything with academics, you know that while it always looks simple, like I'll just send an email and it'll work out, it is never simple. People do not respond. You have to remind them again and again and again. So kudos, RIM, for also being so patient with all of us.
So initially, I was really proud of RIM for taking on all these responsibilities, and citing her as an example for other students to contribute to the community. But then at some point, I told her, "Hey, you have to stop doing this. You're spending too much time on it." So yeah, I'm grateful for the role model. For the role model you've been for so many students here, and she's still being able to be producing good research, and she's, I think, an example for students here in many ways. And thank you.
That's why I didn't know.
I think the lab reps will also say words.
Yeah.
Little present for RIM.
That's a big last day. Sorry. Like your close friends that you know at Mila that's already left, and also the students community. So thank you for everything you've done for us.
And I feel like it's also a sign that now we have 4 organizers. That speaks to how much that work that is. Like it was just RIM for 6 years, and now it's 4 of us doing the same job. And only now that I'm an organizer, I realize how much service it was. So thank you.
And let's not forget to thank Brendan again. Thank you, Brendan.